Welcome to The Explainer. Today, we're diving into a real scientific puzzle. Imagine a pill that, at high doses, is basically poison to our cells, but at super low doses, might just hold a key to slowing down the aging process. Let's get into it. So the drug we're talking about here is called rapamycin, and it has this kind of split personality. On one hand, it's known to be toxic, stopping cells in their tracks. But on the other hand, a ton of research shows it can actually extend lifespan. So what's the deal? And this really gets to the heart of the matter we're exploring today. How can the exact same substance have such completely opposite effects? How can it be both a powerful medicine and a dangerous poison? I mean, just look at this. The difference is night and day. The study we're digging into confirms that at high doses, rapamycin is cytotoxic, which is just a fancy way of saying it's toxic to cells. It triggers cell death and stops them from growing. But at these very low doses, it flips the script entirely. It protects those same cells from aging and actually makes them more likely to survive. Okay, so how do we explain this? How do we solve this paradox? Well, it's not a mistake in the research. It's actually a fundamental principle in biology. Turns out it's all about finding that just right sweet spot. And that principle has a name, hormesis. It's this fascinating idea that the dose doesn't just make the poison, it can also make the medicine. You know, think about exercise. A little bit of stress on your muscles makes them stronger, right? But a huge amount of stress can cause a serious injury. That is hormesis in a nutshell. And this chart just lays it out so clearly. The study this explainer is covering showed this exact thing in cells that are a lot like our own liver cells. You can see the low doses actually boosted cell viability, their ability to survive, by about 20%. But the higher doses, they caused a 30% drop. It's the perfect Goldilocks principle. Not too much, not too little, but just right. All right, so we get the what. Low doses are good, high doses are bad. But what about the how? What's actually happening inside the cell to cause this? To understand that, we need to talk about a critical master switch that controls pretty much all cell growth. That switch is a protein called mTOR, and you can seriously think of it like the gas pedal for your cells. When mTOR is active, the pedal is to the metal, and the cell is in growth mode. Rapamycin's whole job is to interact with that very pedal. And here's where it gets really interesting. It's all about how hard you hit the brakes. High doses of rapamycin basically slam the brakes on mTOR, shutting it down completely. This fires up other signals, like the protein P53, which is kind of like the cell's self-destruct button. But these low hormetic doses, they just gently ease off the gas, only partially inhibiting mTOR by about 40 to 50%. And that sends a completely different and much more beneficial signal. And that gentle signal, that easing off the gas pedal, that's where the anti-aging magic really begins. It has a huge effect on the part of our cells that we most associate with getting older, our mitochondria. Right, we all remember from biology class, mitochondria are the powerhouses of the cell. They generate all the energy. But the source material for this explainer also calls them the biological clocks of aging. As they get damaged and start to break down over time, the whole cell ages. So if you want to slow down that clock, you've got to keep your mitochondria healthy. And what the study found is that this partial mTOR inhibition kicks off an amazing cleanup and rebuilding process. It's like a full-service tune-up for your cell's little engines. First, it clears out all the old, junky mitochondria. That process is called mitophagy. Then, the cell gets the signal to build fresh, brand new, healthy ones, a process called biogenesis. The researchers even figured out these specific proteins that act as the maintenance crew. To handle the cleanup, the mitophagy, the cell brings in these proteins called PINK1 and PARKin. Then, for the rebuilding, the biogenesis, it calls on a team that includes PGC1 alpha and YY1. So low-dose rapamycin is basically the foreman telling the cell to get this expert crew on the job stat. Now, this mitochondrial tune-up doesn't just happen in a vacuum. It kicks off a chain reaction, this incredible cascade of positive effects that just ripples through the entire cell, pushing back against all those hallmarks of aging. And the results are exactly what you'd hope to see from an anti-aging therapy. With healthier mitochondria, your cells have less oxidative stress and less damage to their DNA, their membranes get stronger, they age slower, and they grow in a healthier way. And maybe the most exciting part, the study found a colossal increase in a protein called SIRT1, which is famous in longevity circles as the anti-aging molecule. And hey, don't just take my word for it. The researchers themselves summed it up perfectly. They concluded that this gentle, hormetic effect, that just right dose, directly creates a positive anti-aging outcome because it improves the conversation between that mTOR master switch and the mitochondria. So let's just zoom out for a second. Why does this one study matter so much? What's the really big takeaway here for how we think about aging in the future? At the end of the day, the real insight here isn't just about rapamycin. It's about this incredibly powerful concept of hormesis. This research really suggests a huge shift in our thinking. Maybe we should move away from looking for simple on-off switches in our biology and start looking for the dimmer switches that let us fine-tune ourselves for better health. And that leaves us with a really provocative question for the future of medicine. What if the key isn't always about discovering some brand new, shiny drug, but about figuring out the just right dose of the incredible tools we already have? Something to think about. Thanks for joining this explainer.